paper today on the subject of nutritional genomics. And the idea behind nutritional genomics is to examine the genome of someone and to see whether or not a mutation in that gene would affect nutrition and health. And the title of this paper is Personalized Nutritional Genomics as a Potential Tool for Targeted Medical Nutritional Therapy. And it was published in 2007 by Valkili and 14 other authors. It's a systematic review of 140 other papers. Of this experiment is to see whether or not, by looking at an entire population, if you can find an association between genetic mutation and health outcomes. They did this by looking at specific single nucleotide polymorphisms called SNPs, which code for specific enzymes which function for important health functions. So they looked at 4 different polymorphisms. And the idea being, if you have a mutation in a codon for a protein, then it's going to change the shape of the enzyme and lower the functionality because it's not going to be the right shape. So that you can thus assume that having a poorly functioning enzyme will have a lower concentration of the product. So if your enzyme is its function is metabolizing an important health nutrient like folate or high-density lipoproteins, then you could say a mutation in that gene would lead to lower levels of folate, and lower levels of folate is associated with increased rates of heart attack and negative health outcomes. So the goal of this study is to examine a large population spanning an age from 5 to 98 and the numbers of the hundreds of thousands of different people all over the world, such that it's a good sample of the population as a whole, and you could see whether or not a mutation in the gene which codes for the enzyme which metabolizes folate would be associated with negative health outcomes. In the first case, they were looking at at an enzyme which codes for the enzyme which metabolizes folate, and folate in the blood. And they found a small but significant increase in association between levels of folate, between this gene mutation and increased rates of cardiac arrests and negative health outcomes. And the second study, they found that a gene mutation in a gene which codes for a protein that's part of the chylomicron in high-density lipoproteins is associated with lower rates of high-density lipoproteins, and thus also higher rates of negative health outcomes due to lower levels of high-density lipoproteins. In the third gene mutation, this was also the case. It was also a different part of the chylomicron which is mutated, and then thus it should lead to lower percentage of high-density lipoproteins. And the evidence suggests that there is a small but significant association between this gene mutation and lower levels of high-density lipoproteins and increased negative health outcomes. In the fourth case, it was a gene mutation in the enzyme which helps break down low-density lipoproteins into medium or high-density lipoproteins, so that the mutation in this gene would lead to a lower functionality in the gene in the protein which lowers the level of low-density lipoproteins, such that there's an increased level of low-density lipoproteins. And this turned out to be the case. There was a small but significant association between the low-density lipoproteins and increased negative health outcomes. Now, problems with this study is that it's a systematic review. so that you can't find a direct correlation because you can't look at the specificity of the different studies. It was not given the information about every single specific study of the 140 was not provided. So you can't make a specific claim. But it does provide a huge sample of the population. So it suggests that there's good evidence that there is something there, that the single nucleotide polymorphisms are associated with mutations in the enzymes and thus lower functionality of those enzymes and negative or positive health outcomes. So it's a good suggestion for further areas of study about nutritional genomics. Thank you.